Okay. PowerPoint to record, not to the crutch. Okay. Let's see. We finished halfway through that first lecture, so I'm going to start where I left off and kind of go through the second half kind of fast because I've already talked about a lot of this. So the first part of that last lecture was design considerations. Things you need to think about before you start your experiments. Second part of that lecture is assembly considerations. So you already know what you're going to build, how you're going to deliver it, how you're going to microinject it, etc., blah, blah, blah. Now you need to figure out how you're going to build it, the assembly. So one of the things you need to think about, and I'm going to go through these quick so I can move to the next lecture, is which organism are you going to use? Are you going to use E. coli? Or are you going to use yeast? Or something else to assemble your designed constructs? Now, you're going to want to use E. coli if most of your things are small. E. coli is really good for really, really small stuff. Yeast is good for assembling really, really big things through recombination. Good morning. Okay. And we got to remember that the E. coli doubling time, what did I say it was? 30 minutes. Yeast was 90 minutes. So anything you do in yeast is going to take 3 times longer. But sometimes, if you're doing big stuff like greater than 20,000 base pairs, sometimes yeast is just the way to go. Okay. So think about how big the final thing you're building is going to be. And that might help you decide what you really want to build it in. Okay. Now here's one thing we haven't talked about yet is a lot of people nowadays, they're not even building their own constructs. They're synthesizing them. So I just want you to know that that is an option. So what that means is you can go to a company like GenScript, and GenScript has large machines that can write your sequence. So as long as you know your sequence, you have it designed out, you can give them a sequence. Let's say it's 500 base pairs long. It's a little tiny gene. You can give them the ATG to the stop codon, and they can build that plasmid for you. And sometimes that's faster. Sometimes that can be cost prohibitive. When you start to build things that are greater than 3,000 base pairs long, it starts to get expensive. It starts to get to cost like $3,000, something like that. And when you start doing things that are like 5,000 or 10,000, it can get expensive. So, but sometimes if you're doing something tiny, if you just need like a little construct that's 100 base pairs long, and it would in theory take you 3 steps to assemble this thing through restriction enzyme cloning, GenScript could just synthesize that for, I don't know, maybe like $250. So sometimes it's faster to just literally go to a company and say, this is the sequence I need, build this for me, and order it. And you got to consider this, especially when in the lab, what's really expensive is your time, right? Paying people to make things is really expensive. So consider this as you are asked to build things. Sometimes it's actually just cheaper to have a company build it for you because they can do it with a machine. Okay, the other thing you need to think about, and again, this relates to size, is the quantity of information. So that literally means the base pairs. How long is it? And that's going to determine, and we've talked about how the size of things can balloon. If you are working on a vector for Drosophila, UASP, ATTB, it's got ATTB site, it's got a white gene, it's got an antibiotic cassette, it's got a promoter, sometimes they're long, it's got a multi-cloning site. If you also inserted something in there, it's got that, it's got a terminator. These things get really long. So you got to always know the size of the things that you are planning to build. Let's see. Let's move on. Let's move to the physical assembly. So how you build these things. And we're going to have a lecture on each of these things in the next module. So one thing you can do, which we started off in the lab, started planning out, is restriction enzyme cloning. And we're going to go through a protocol in detail on how to do that. Okay. So that's when we're using restriction enzymes. What's a restriction enzyme? Yes, it cuts DNA. And why is it restricted? No, that's DPN1. DPN1 is a restriction enzyme. That is correct. But why is it restricted? Yes. It recognizes a site. Remember when we planned out those primers, we used BAMH1, which was GGATCC. And if you map that out on the other strand, GGATCC, I told you that it reads the same forward as it does backwards. This is called a palindrome. Reads the same forward as it is backwards. And restriction enzymes recognize these palindromes and cut the DNA like that. So that's restriction enzyme cloning, and you build things by sticky ends. Sticky ends. Okay, so we'll talk a lot about that. Good morning. Gibson cloning. So there's Gibson cloning. What else is there? There's sewing. We're going to have a whole lecture on these. These are techniques that allow you to, what you would say, stitch together DNA pieces with homology regions. Okay, so we'll have a whole talk about how this works, but it's not restriction enzyme cloning because you don't use restriction enzymes. You basically just use a polymerase to write regions of homology, and then your regions of homology can bind to each other, and you can, through PCR, so this is a PCR-based technique, you can stitch together pieces. So I'll just draw it out just quick, just a little brief sample of how this works. So imagine you have piece A and you have piece B, and you want to make these things stick together. You want to fuse A to B. So what you can do is you can design primer here, primer here, primer here, primer here, and then you make this primer have a homology arm that matches right here, okay? And you make this primer match that, have a homology region that matches here. Now when you amplify these products, what do you get? You get something that looks, I use bad colors. You get something that looks like this. You can fuse things together. Now I just did this quickly. I'll map it out exactly how you do this in a whole lecture. So I don't want to get into too much detail, but the concept that I want you to know now is that you can assemble things with restriction enzymes. You can assemble things with PCR-based methods that stitch together DNA based on homology arms, homology regions. And then we've talked about homologous recombination, which is another homology-based assembly method, but it's using sometimes enzymes within a cell. Okay? So we're going to talk about all those different ways to assemble things. And then we also said you can also synthesize constructs from a machine. Okay. So that's the end of that first lecture. Let's go into the second lecture, and we'll slow down a bit here. Okay. So I want to talk about some of the common problems and troubleshooting that you will encounter. So and I also want to talk about some tricks. Okay. So let's first go back to where we left off at the lab, and let's talk about some tricks. So let's say we had that one gene that we wanted to clone, geneX, right? And we designed primers like this, and on one primer was 5' end, we put BAMH1, BMH1. And on the other end, we put Jo1 on the 3' end. And then we also added little random overhangs so that the enzymes could grab that and cut. So our strategy was to PCR this product, digest it, also digest a plasmid, where our plasmid would have had BMH1 and Jo1. And then these sticky ends would match here and here, and then we could ligate that. That was our strategy. Does that make sense? Okay. So here's a question. Let's talk about a trick. Let's say we don't want to just clone geneX. We want to clone his6 geneX. How do we do that? What do you guys think? Where would you precisely put it? And how would you, that's correct, we want to add that sequence in front of geneX. But how would you do it? How would you build it? Right? That's right. We want to put it here. But how? Like literally how? How do we do that? Any ideas? How can we add a sequence that's not there? Was the BAMH1 site there before? No. How did we add the BAMH1 site? We literally just stuck it on our primers. And when we ordered our primers, it was in there, right? So that means when we design our primers, okay, so let's say we have the forward primer. Let's say it was A, T, G, X, Y, Z, blah, 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 to here. Here was a final G. That was our CG clamp. So let's say this was our forward primer. And we added G, G, A, T, C, C, A, 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 A. We just stuck that on our primer. And so when we ordered our primer, we weren't going to just order this. We were going to tell them this. And remember I said, this is kind of like a floppy thing. And this is what will actually anneal. Okay. So if we want to add a tag, all you got to do, I mean, this sounds simple, but sometimes nobody teaches people this. All you got to do is literally right after your restriction site, what do you want there? You want the start codon. You want cat, 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 cat. And then your primer starts. Okay. And this is going to be what's annealing. And this is just going to be kind of like hanging out. But what gets PCR'd? What's going to get PCR'd? All this is going to get PCR'd. So you're going to get a PCR product that is geneX. You'll get 1 million copies of geneX with all this sequence built in. And then when you do the restriction digest, you'll cut off a little bit at the ends to make the sticky ends. But you're going to have that his tag and that start codon built in from your primers. So the key thing is here is that you can build sequence into your primers. That's literally how you can, anything you can think of, you can add that to your primers, and you can get primers that are hundreds of base pairs long. Okay? So you are not limited. You're only limited by your brain, your big brains. You got to use them, and you got to design things. Okay? And you can just put those onto your primers, and then when you do your PCR, you're adding that sequence into your primers. Okay? Sometimes you can actually even order oligos that have homology regions, and you can literally just synthesize your own construct. Okay? So we can just put in the sequence that we want into the primers when we order them. Okay, so what is the catch with that? What's the catch? What did we learn as we had our primer, 5', 3', and then what happened when we added the restriction site? What happened? It might have matched, and it would fold back on itself. So what's the catch? The catch is the longer primers are, the less likely they are to work. Okay. So you can do things, you can do anything you can think of, but things get harder as your primers get longer. Okay. And sometimes when you have hairpins, sometimes when you're working with long primers, you cannot avoid creating hairpins because there's so much sequence and there's only 4 base pairs. There's only A's, T's, G's, and C's. And if you have a sequence that's 50 base pairs long, just by random stochastic randomness, there's going to be a site where it can hairpin. Okay. So the next question is, we want to use these tricks. We want to put anything we want on our primers, but we know we're going to make hairpins. So how can we avoid hairpins? How could we do that? How would you get rid of a hairpin? So let's think about it. What is a hairpin? A hairpin is hydrogen bonds binding, annealing to each other, right? That's exactly what it is. How do you get rid of hydrogen bonds? If you heat it up, right? So there's a technique called hotstart. Hotstart PCR. So let me walk you through what's happening. We've talked about the PCR reaction. PCR reaction is just a loop of denature, which is heat, anneal, which is your annealing temperature of your primers. And that's where you cool to create the binding, the hydrogen bonds. And then there's extension. Okay. Now, here's a problem. Here's a problem with this. The first step, the thermocycler, the machine, so here's the machine, thermocycler. The thermocycler starts cold. And when you tell it to start this loop, the first thing it does is a 98 denature. But does it start at 98 degrees Celsius? No, you put the tubes in and it starts at 23 degrees Celsius, which is room temp. And then it ramps up to that temperature. So there are problems that are induced by starting at room temp and ramping up. What could you think of the problem might be? Well, let me show you. Let's say you have a hairpin like this. You have a long, long primer and it hairpins at the end. You mix that tube of PCR, you put it in your thermocycler. As soon as you add, what's fusion polymerase? It's the writing. It's the high fidelity writing function. As soon as you add that, what's going to happen? As soon as you add the fusion, what will happen? It's going to start writing right away. Okay. And what's it bound? What's annealed right now? Has your primer annealed to the template yet? No, but it's annealed to itself. So what's going to happen? Your fusion polymerase is going to destroy your primers because it's going to write off the hairpin. Does that make sense? So you've just destroyed your reaction. So how do you solve this problem? If the problem is that you're not hot enough early, you do a hot start. Okay. So a hot start is, let me tell you how you do the hot start. This is how you get rid of and avoid hairpins. And you should actually just do this every time you do a PCR because it always works better. So here's how you do a hot start. All you need to do is when you mix your tube, of your reagents. What goes in there? What goes in that tube? DNA. Your template DNA. What else? Your primers. What else? Your DNTPs. What else? A buffer. Water. And yes, but we don't want to add diffusion right away. Wait on the fusion. Wait on the fusion. Normally you can, normally it's going to work. If you don't have to worry about hairpins, just throw the fusion in and ramp up the thermocycler. But if you're worried about the hairpins, you wait on the fusion. You mix up this, you put your tube in the thermocycler, you start the program, and you program it after the 98 2-minute initial denature, where you're just denaturing all the DNA. You cool it down to 85. 85 is still hot enough to make sure everything is denatured. Does that make sense? So everything is hot. It's not your hairpins. If you had hairpins, they're no longer hairpins because it's denatured. Okay? And then you let it sit let's say you just set that temp, or you just set that time for 30 seconds. As soon as that step hits and the thermocycler hits 85 degrees Celsius, you push the pause button, you open the lid, you pull out your tube, and you quickly add your polymerase. Close the lid, put it back in, close the lid, press the pause button. Now you're playing. Now you're good. Now it's going to ramp up from 85 into your loop. And then it's going to loop and you don't have to worry about your hairpins. Does that make sense? Okay. So hotstart PCR is a way to get rid of those hairpins. And if you really start building stuff, almost every primer you design will have hairpins because it's impossible to avoid not having them. So get used to the hotstart. Okay, so let's go through a troubleshooting. What's the most common problem with PCR? You design primers, you do your PCR, and you get nothing. It doesn't work. So what do? What would be the first thing what are some ideas?
Change your primer.
Okay, fundamental rule of science. Tattoo this on yourself. Check your positive and negative controls. That should be the first thing you always do when something never works. And things will never work. So you always need a positive and negative control. So you check so if you were going to do a PCR, and you are testing new primers, I would run a PCR that you know is going to work. You've done it before. You know this thing's going to work. Okay, so that's a control for your mix. Maybe you forgot to add the DNTPs. Shit happens when you're mixing stuff. Maybe you were distracted or you're listening to your iPhone or you were Skyping with somebody in the lab, which happens. Maybe stuff happened. Okay, so you need a positive control that you know will work so that you know you didn't just make the wrong mix or something like that. Okay, that's going to check to make sure that just you set up your mix right, you programmed your thermocycler right, everything was working. So your positive control should be like that. If it's not there, if your positive control band isn't there, then you know like you messed up mixing. It was your fault. It was your fault if your positive control band is not there. Okay? Minus control is going to be just testing for contamination. So if your minus control has a band, what are you going to worry about? And you see a bunch of bands in your tests. You're going to worry that this is not actually what you wanted to PCR if your minus control has. So oftentimes the most common minus control in PCR is you set up a mix, but instead of adding template, you add water. Water's got no DNA in it, so it'll be a negative control, right? And if there was contaminating DNA, you'll see a positive band in your negative control, okay? So the first thing you need to check is that your positive control worked and your negative control did not work. That's what you want, okay? So let's say that's good. You check your positive, negative controls. That was step one, okay? What's the next thing you do? Where could the source of the problem be? Well, just think about the reagents. What are the reagents again? Let's look up top at the reagents. You guys said DNA, primers, DNTPs, buffer, water. Which is the one thing you controlled? Which is the one thing you probably messed up? Your primers. If you guys are newbies, you probably just messed up your primers. Okay. So second thing is, and primers can be confusing, right? There's two directions of DNA. Maybe you had a brain fart and you did not reverse orient your reverse primer or something like that. Maybe you made a mutation or maybe you made a typing error. Something happened. So check your primers. Check primers. And when I say check your primers, you pull up. This is why when you design your primers, you design that Word file and you design that logic. You go back to that document. You look at your template, which you mapped out in FASTA format. What's FASTA format? It's literally just like the sequence written out with a little caret here. Caret is that. That's FASTA format. So when we made that design document, we pulled the gene off the database, we pasted it. That was in FASTA format. So check that. And what you want to check is, do your primers that you designed, do they actually match that sequence here? Does your forward match that sequence here? And does your reverse complement primer match the reverse complement of this sequence right here? Does it match perfectly? Okay. Maybe if you forgot to check for hairpins, maybe you want to go back and recheck for hairpins. That happened to Dylan. He designed a primer that had a hairpin, and all we had to do is get a different primer with a new hairpin. I think it worked. Is that correct? That's correct. So the most common problem and the easiest thing to fix is your primers. Okay.
So hotstart does not negate all hairpins.
I would say yes, it does negate all hairpins because heating anything is going to denature those hydrogen bonds. It's not going to denature the hydrogen bonds, but it's not going to ensure that your PCR reaction will work. Does that make sense?
Yeah.
Some things just don't work, but usually hotstart will overcome most problems with hairpins. But there are lots of other issues with primers. So, okay, let's just talk about them. Let's talk about other problems with long primers. You got a real long primer. The only thing that's annealing is right here. Okay? All this stuff can potentially anneal to other things. So if your template is impure, and by that I mean, say you did a genomic extraction from an insect. Okay. You have a tube that's full of the entire genome of that insect. Is it likely that there are other regions in the genome where these primers can anneal to? Yes, it's very likely. The longer your primers get, the more you increase the chance of off-target amplicons. So you can control this by having a really, really pure template. So sometimes the easiest thing to do is to first just clone your gene into the gateway plasmid, right? Clone the gene, just the gene, into the gateway plasmid. And that way your primers can be very, very specific. You're not adding on other stuff. And then maybe you do a step 2 where you subclone it into a different thing and you use this plasmid, which can be highly purified. And so then your template is highly pure. That's all that's in the sample. So then you don't worry as much about long primers if your template is highly pure. So template purity is another thing you worry about, you think about. The more pure your template is, the easier it is to get complicated PCR reactions to work. And the dirtier your template is, the harder it is to get complicated PCR to work. Okay, third thing. So you check your primers. You had your positive control. They were good. You checked your primers. You didn't do any logical errors in your primers. They should be perfect. Okay, what's the next thing you do? Well, the next thing, what's the other thing in the PCR that you did? So you chose your annealing temperature. You chose that. We put that into a little computer program and it spit it out and it said salt adjusted temperature 56 point whatever, right? And then we just programmed the annealing to that. Maybe that's not actually the best annealing temperature for your primers. Maybe there's something happening and maybe a different temperature is going to make things work. So the third thing that usually fixes the problem is adjusting the annealing temp. Okay. So if the first thing was redesigning your primers, oftentimes I'll tell you the order of how I do things. I'll check my primers. I'll check my positive and negative control. I'll check my primers, and then I'll try a new PCR reaction with what's called a gradient PCR. So let me talk about what a gradient PCR is. If here's your thermocycler, lid up. Thermocyclers will have rows that you can insert the tubes in. Have you guys seen these before? Thermocycler, yeah. No, okay. Well, they have this little metal block, and in this little metal block are these holes that you stick the tubes into. And the role of the metal block is to heat and cool to the temperatures. That's how the functionality of the heating and cooling loops works, is it heats this metal block. Okay? So you can do, in many of these thermocyclers, these are called thermocyclers. Why are they called thermocyclers? Cycles the temperature. That's what PCR is. It just cycles, it just loops temperature. So these are thermocyclers. Now what you can do is you can program the machine to be 60 degrees Celsius here. Actually, I should make this red. 60 degrees Celsius here and 50 degrees Celsius here. And every row in between is an increment. 51, 53, 54, 55, whatever, up to 60. Does that make sense? This is a gradient, a temperature gradient that goes from low to high. And what you can do is you can mix up 1, 2, 3, 4, 5, 6, 7. Let's say you had 7 rows. You can mix up 7 tubes of the same mix. So you mix one tube, you aliquot it into 7 little thermocycler tubes, and you put one here, one here, one here, one here, one here, one here, one here. Okay? So you're doing the same PCR, and the only thing you're varying, what's your variable? The temperature. You're checking different annealing temperatures, okay? So you do your gradient PCR, and then you pull these out, and you run your agarose gel, got your positive, got your negative, and then you got your 1 to 7 increasing temperature. And sometimes what you'll see is what would that suggest? This is the best. And then you pick that temperature. And then from then on, you only do PCR with those primers at that temperature. Does that make sense? So the third thing you can do to fix no amplicon is change annealing. Sometimes this will fix your problem. Okay, so here's my workflow. You'll be doing PCRs every day if you're quick and good at this. And what you do is, a lot of times these just don't work. And so what you do is, first, the workflow, just like I said, to repeat, check positive, negative. If that doesn't work, check the logic of your primers. If that doesn't work, do a gradient. If that doesn't work, you throw your primers away and you order new ones. Sometimes things that you programmed into that sequence just don't work. And oftentimes you can fix the problem just by designing new primers. Just change the sequence. And if I go through this workflow and it still doesn't work, then I go to a new cloning method, or I synthesize. Does that make sense? Don't bang your head against the wall forever. Be persistent, go through the workflow, but if it doesn't work, then go to a new cloning method or synthesize or pick a new project. Okay? So that's how you troubleshoot cloning. And the first step of cloning is PCR, and that's usually where people mostly fail. Okay, so let's walk through some. You can slow down. I got time. Okay, let's walk through restriction, enzyme, digest, cloning, data. Okay, so we make our plasmid. The step after is to verify that we actually made that plasmid. We have to verify that through data because we can't see it with our eyeballs. We can't see the sequence. So we have to verify that the construct we made is really what we think it is, okay? So one of the ways that we do that is restriction enzyme digest verification. I've talked about this before. I'll reiterate it again. So if we clone geneX with A here and B here, we can do an agarose gel. What would our plus control be? Our plus control could be, in this case, I would say we would just quickly do a minus control. The minus control is going to be our empty backbone. What's the backbone? The plasmid. Let's say this was P bad. So we just put PBAT in here. And PBAT is about, so here's our ladder. PBAT is about 4,000 base pairs. So our minus control is going to look like this if it's digested. If we digest. Because we linearized, so we don't have the 3 bands because we digested, right? We got that. Okay, and then our test is going to be, we're going to expect to see this. And let's say our geneX is 1,000 base pairs, we're going to expect to see something down here if we digested with A plus B. Okay, so let's show you. I'll show you how to actually like do those data sets. Let's see if I can